Hershey and Chase did an important experiment because at that time they didn't know what is the genetic material. Is it DNA or is it protein? I mean, now we know it's of course DNA, but they have designed an experiment to figure this out, and I'm gonna just explain this experiment to you, okay? So first of all, there are a few things you have to understand. You have to understand that DNA contains phosphorus, okay, as an element. Yeah? Phosphorus, and protein contains an element called sulfur, okay? And basically what they were asking is when they used viruses to do that, and they're now asking, is now phosphorus passed on? Or is sulfur passed on? And if phosphorus is passed on, then we know that DNA is passed on to the next generation. And if sulfur is passed on, then we know it's the protein. So that's kind of the bottom line that they were trying to figure out is we can distinguish between DNA and protein by looking at the phosphorus or the sulfur that is in them. Okay. And so what they've done is the following. They've taken bacteria, and I'm just going to draw a bacterium over here. And they've taken a virus, it's the so-called the T2 virus. That's simply the name of the virus. It was called the T2 virus. And what they have done is, and this virus looks a little bit like this, okay? And in this virus, it looks a little bit, it has these legs sticking out. And in this virus, we have the genetic material, right? But they didn't know, is it DNA or is it protein? That was kind of the question, yeah? And on the outside over here, we had protein, because the virus itself is made of protein. Okay, and what they've done is now the following, okay? They wanted to know is now the DNA, when the virus goes into and infects the cell, does it now pass on protein or DNA? That's essentially what they wanted to know. And if the virus injects protein, then protein must be the genetic material. If it injects the DNA, then the DNA is the genetic material. And we can figure this out by determining or by having a look at whether phosphorus or sulfur is passed in, right? So what they have done is the following. They've taken the virus. I'm just going to make a bigger drawing over here right now. It's actually how it looks like. Yeah. And they have the protein on the outside. They have used radioactive sulfur. It is possible to do that. Okay, it is possible to have radioactive sulfur, and this is sulfur 35. Okay, because it's the protein. And on the inside, they had their DNA, and this was phosphorus, and the phosphorus was phosphorus 32. Okay, so that's the DNA. Okay, and the sulfur, that's the protein. Okay, and what they've done is the following. They have now mixed the bacteria with the virus, and now the virus attached to the bacterium, because that's what they do, like this. And they have, and it has injected some material, and in this case, they wanted to now know what is it that is injected. Is the phosphorus injected or is the sulfur injected, right? Is the DNA or the protein transferred or injected into the cell? And what they have done is the following. They gave the cell a few, mix the cells, the bacterial cells and the viruses for a few minutes, and then they have shaken it up. They have, they've shaken it, and this caused the virus to separate again, because it kind of broke off, right? So the virus was kind of broken off. Bad drawing, okay? It left, yeah, it was went away, and we had now some stuff in here. Right, okay, so and now they were kind of looking, what do we have in here? Is this now radioactive phosphorus that we have in here, and then it's DNA, or do we have radioactive sulfur in here? Then it is protein. So they have had the whole thing here. All of this mix was in a little test tube. I'm going to draw this test tube, okay, and they have done and they're floating around. Everything was mixed to break the whole thing off, and then they have been spinning the whole thing in a centrifuge. The centrifuge it spins very quickly, and this causes the cells, the bacterial cells, to build up at the bottom over here. It kind of sinks down very quickly, and the viruses are here. So these viruses over here, the viruses, these guys over here, they're up here. That's the T2 virus, and over here we have the cells, the bacterial cells, with some stuff in them from the virus. Okay, and now what they found out that the T2 virus here, the liquid here, the top liquid over here, this contained sulfur 35. So this means that over here, this contained the outside of the virus, the protein, that's the protein, and the cells, they contained phosphorus 32 from the virus. Yeah, the cells contain phosphorus 32, yeah, down here, and this means that the DNA in the cell, which we found down here, contains phosphorus 32, and this basically means if we know that the cells now contain the phosphorus from the virus, then we now know that it must have been DNA, because DNA contains the phosphorus. Okay, so this is how they figured the whole thing out. Okay, by determining whether phosphorus or a protein is passed on. And now you might wonder, why do we have to use radioactive phosphorus or radioactive sulfur? Because, well, otherwise you're not able to see it. Oops, that's a previous foil. That's a little bit, okay? Otherwise you're not able to see this, okay? So you have to label this. You cannot simply sulfur and phosphorus. It doesn't have, yeah. You cannot simply know it by looking at it. So you have to somehow label it, and they've labeled it radioactively. Okay. So this is the famous Hershey and Chase experiment, and they were the guys who figured out that it is DNA that is the genetic material and not the protein. Okay. It's a very elegant experiment, but a very powerful one. That also illustrates how radioactive labeling is used to distinguish between the different macromolecules like proteins and DNA. Okay, so that is it.